All right, today we are getting into cellular respiration. Now, one thing to keep in mind is that, only plant cells can do photosynthesis, as you know, but all cells, plant cells, animal cells, both kinds, need to do cellular respiration. Okay? So basically what we're looking at here is we are looking at, okay, when you eat food and you're breaking it down, what's actually happening with it eventually. And the eventual endpoint is cellular respiration. Okay, so things keep getting broken down, into smaller and smaller pieces. Okay, and as those bonds are broken, energy is released and some is lost to heat, which for us are endotherms, right? That's good for us. It helps us maintain our body temperature. Okay, and basically, when it comes down to it, the fuel that our body is using for most of its functions is glucose. So that's the end product of a lot of the macromolecules as it keeps getting broken down would be glucose, okay? Especially here carbohydrates because of course glucose is a sugar, okay? So that's why carbohydrates are good energy molecules because we end up getting glucose out of the mix. And cellular respiration is basically the process in which we are taking different things, breaking it down into, breaking that glucose apart into its various pieces for energy. And this is the most efficient metabolic pathway. And it happens to be catabolic because we're breaking something down. Here's the reaction for cellular respiration. This is in the presence of oxygen. Later on, at a different point in time, we'll talk about what do cells do when they don't have access to oxygen, but this is oxygen. And this should look really, really familiar to you because this is the exact reverse of photosynthesis, okay? So here, instead of making glucose and oxygen, we're taking glucose and oxygen, we're splitting them apart into carbon dioxide, water, and we are releasing energy instead of needing the energy input. Remember, in photosynthesis, this energy input was light energy from the sun, okay? So basically what we're doing here is we're taking glucose that's made by plant cells, which you get either if you're a plant cell or you get by eating a plant cell or eating a thing that ate a plant cell, okay? And we're going to take that glucose, we're going to break it down and make energy in the form of ATP, and that ATP we're going to use to do all kinds of cellular work like we learned about before, transport work, mechanical work, chemical work, any of that stuff. So the mitochondria is the powerhouse of the cell, as you all know, and that is because cellular respiration happens in the mitochondria, and cellular respiration produces ATP, okay? And so specifically, part of cellular respiration is happening right surrounding the mitochondria in the cytoplasm. Part of it is taking place here, right in this, where this like yellow inner membrane is, okay? There's a phospholipid bilayer right here. Part of it's taking place right there, okay? Part of it's taking place in this blue area inside the matrix, okay? And so that is kind of where this is all happening in our actual mitochondria. Similar to photosynthesis, we're going to do a brief overview here in the online lecture, and then in class we'll diagram this. We'll get a little bit more into the details of what you need to know, but in general, cellular respiration is 4 steps, okay? Glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. I know, all of the words, all of the vocab, don't get too scared by it, okay? But lots and lots of vocab with this unit. Step 1 here, glycolysis. Look at the name, it should give you a hint. Lysis means to break apart. Glyco is going to come from our glucose. Okay, so we're breaking apart glucose here. So as it turns out, we split it apart into 2 different sugars called pyruvates. Okay, this ends up taking a little bit of energy, but it also ends up producing energy, and so we end up ahead to ATP. It also makes this stuff called NADH, which we'll talk about later, and this is happening in the cytoplasm right around the mitochondria. Now, I'm glossing over a lot of stuff because this is actually all of the reactions that take place to get this pyruvate, but you don't need to know all this. What you need to know is, hey, start with glucose and with pyruvate, and along the way, we got some ATP made and this thing called like NADH made, okay? Cool. Next is the oxidation of pyruvate. So here's that pyruvate that was made, and now some things are going to happen to it. So the pyruvate is going to enter into the mitochondria, into the matrix, the innermost zone, okay? Through active transport, it's going to get pumped in, okay? And basically, there's a whole lot of reactions that happen, and this is going to release carbon dioxide, and it's going to make this little thing called NADH, which is basically an electron carrier, okay? That's what reduction is. So we're going to add some electrons to this NAD plus to make NADH. And then basically all of the leftovers are combined to form this acetyl-CoA thing, okay, which is just going to be part of our next step. And here's what's actually going on. Again, you don't really need to know. Basically, what you need to know is pyruvate ends up becoming this NADH2, this carbon dioxide, and you get this acetyl-CoA left over. The CoA stands for coenzyme A. And next is the citric acid cycle, which is sometimes called the Krebs cycle, depending on what textbook or what resource you're using. And basically here, acetyl-CoA splits, the enzyme goes away, we release more carbon dioxide, we make some more NADH and some FADH2s. Again, these are just little electron carrier molecules. We also make a couple ATPs. This ATP is made in a different way that we've seen before. This way is called substrate level phosphorylation. And so basically what's happening here is here we've got some type of molecule with a phosphate on it, and here we've got an ADP. And so there's going to be an enzyme, here's the active site, that's going to take this phosphate, snatch it off of this guy, and plop it onto ADP to make ATP. And here's our molecule minus its phosphate. Okay, so this is substrate level phosphorylation because this is a substrate, and we're getting the phosphorus from a substrate. Okay. All right, here's the actual cycle. Whoa, okay, you do not need to know any of those steps. We just need to know, we start out with pyruvate, and what we end up with, of course, is some ATP and some carbon dioxide and some electron carriers. And then lastly, we have oxidative phosphorylation. This is kind of sort of a very similar cousin to what happened in photosynthesis with the electron transport chain. Okay, so here's that membrane inside your mitochondria here. And so basically what's going to happen is our NADH and our FADH2 are going to input some electrons into this huge electron transport chain. So they're going to go on a wild ride. And when they do at the end of this wild ride, they're going to meet up with some oxygen and some hydrogen ions, and they are going to combine back into water. Remember, this is the reverse of photosynthesis. And similarly, because we've got these electrons going on here, that's going to cause some hydrogen movements. But notice the hydrogen, instead of moving in, is kind of moving out this time. So this is going to pull hydrogen. These minus electrons are going to pull the positive hydrogens out because opposites attract. And so these positive hydrogens are going to flow out of our matrix here, and they're going to build up a high concentration, and so they're going to high to low diffuse back in through ATP synthase. And so this is going to make an ATP exactly like how it did in photosynthesis, except for this time, instead of diffusing out, they're diffusing in. But it's the exact same idea. Okay. Here's it way more in detail. You don't need to know that detail. We'll draw it out simplified altogether. Okay. So basically, we've talked about now 2 different times when we can make ATP from hydrogen going through ATP synthase, going from high to low. And this name is called chemiosmosis. Okay, chemi because it's like a chemical or an ion. Osmosis because we're diffusing from high to low. Okay, so this happens with both photosynthesis and cellular respiration. The direction of travel is a little bit different, and what gets them started is a little bit different, but the end product is you're going to make some ATP. Pretty cool. So this is kind of a nice general overview. So here's our glycolysis happening in the cytoplasm. Next, that pyruvate gets brought in, made into acetyl-CoA. This is happening in the matrix. Here's our citric acid cycle. Okay, all this stuff ends up coming to our electron transport chain, and this is going to make a whole bunch of ATP. So here's the important part to take home. Glycolysis produces a couple of ATP, citric acid cycle produces a couple of ATP, and the oxidative phosphorylation, that electron transport chain at the end, produces 26 to 28 ATP. So the majority of the ATP come from that big old ride the electrons take and hydrogens flowing in through oxidative phosphorylation. It's a mouthful. Okay, so there's kind of the overview of what's going on with that. So again, we're taking glucose, we're breaking it down. Our energy is ATP. We have these other 2 molecules that are essentially electron carriers. Okay. This is happening in the cytoplasm and in the mitochondria itself. Here's our reaction. It's the exact opposite of photosynthesis. Okay. We make ATP through 2 different ways. Through substrate level, where a phosphorus is stolen from a substrate, or through that electron transport chain and that ATP synthase called oxidative phosphorylation. I'm having a hard time saying that, aren't I? One thing I want to point out is just some efficiency. This was a former AP exam when they used to make you do more math with it, so don't super stress about it. But, okay, we can figure out how efficient cellular respiration is. So glucose, theoretically, stores 686 kilocalories per mole of energy. That's how much it can release when you burn it, okay? If you break down ATP, basically every single time you break apart an ATP, you release 7.3 kilocalories, and we end up making 32 ATP from every one glucose. So how efficient is that? Well, we can take 7.3, how much one releases, and times that by 32. So this is how much ATP energy we get from glucose. But we were said that glucose gave us 686. So if we divide those two to get a percentage, we end up getting 34%. So only 34% of glucose's actual energy is converted into ATP energy. And that sounds really, really low, okay? But actually, car engines are 25% efficient, okay? And 34%, this is the most efficient reaction in the cells. So although 34% sounds kind of terrible, it's actually really, really good, okay? Because the rest of the energy is lost to heat, which is, again, good for us endotherms where we're trying to modulate our own body temperatures. Here's some more slides. Sometimes seeing it in different ways can help you out. Always a good idea to take a look at your book. Hey, remember that thing? To look at what they've got going on in there, but don't get too bogged down in the details because we're not getting too far into a lot of them, okay? So here's some general overview things as a little self-check to see if you understand what's going on, which at least once we go through it in class, you should be able to answer all these questions. And we're in chapter 7 in the textbook in case you want to go check there.